Cell injury. Cell damage is understood as a disorder in the structure and function of intracellular molecules and organelles. Please see the picture, which will remind you of the structure of cell. Causes of cell damage are classified into three groups. They may be physical causes, chemical causes, and other causes. For example, physical causes, they are such physical agents as mechanical forces, for example, trauma, extreme of temperature, burns, heat stroke, freezing, frostbites, electrical injuries, ionizing radiation, ultraviolet radiation, chemical injury may be caused by direct cytotoxic effects of chemical substances and by conversion to reactive toxic metabolites. Other causes include hypoxia, biological agents, nutritional imbalances. Actions of damaging agents on cell and extracellular structures. There are two kinds of actions, direct and mediated. Direct is action of damaging agents on cells and extracellular structures. Mediated damage is action of damaging agents through derangement of the extra and intracellular regulatory systems. Kinds of cell damage. They may be acute and chronic, reversible and irreversible, specific and non-specific, total and partial. Reversible and irreversible cell injury. Reversible cell injury. It results in loss of cell function and in structural changes. In reversible injury, the cell can revert to normal condition if the stressor or injury is removed. Irreversible damages are not removed by tension of compensatory mechanisms. Reversible cell injury. You see an example of reversible cell injury. They are seen under light microscope. They consist in cell swelling, in clumping of chromatin, and lipidosis. Sequential development of biochemical and morphologic changes in cell injury. The first to occur are functional changes, when morphologic and metabolic disorders are still absent. After that, disorders in metabolism add to these disorders, and they are followed by ultrastructural disorders. There is a time lag between the stress and the morphologic changes of cell injury or death. Signs of cell injury. They may be morphological and functional. Morphological signs include changes of shape and color, swelling or shrinking of cell, the disturbance of contact between cells, damage to cell membrane, damage to cell organelles membrane. Functional changes include reduction of cell mobility, disturbance in cell division, change of cellular membrane, intracellular enzymes presence in blood, appearance of new functions, changes of biochemical processes in injured cells. Morphologic changes of cells in reversible and irreversible cell injury. Picture A shows normal kidney tubules with viable epithelial cells. Picture B shows early or reversible ischemic injury, which shows surface blebs, increased eosinophilia of cytoplasm, and swelling of occasional cells. Picture C shows irreversible injury of epithelial cells with loss of nuclei, fragmentation of cells, and leakage of the contents. changes in the nucleus of cells may be karyolysis, pycnosis, and karyorhexis. Pycnosis is progressive shrinkage of nucleus when it turns into dense mass of tightly packed chromatin. Karyolysis is progressive dissolution of nuclear chromatin due to action of DNases of lysosomal origin. Karyorhexis, it means breakage of nucleus to many clumps. So here you see the picture of karyorhexis, fragmentation of nucleus. General mechanisms of cell damage include: first, disorders in energy metabolism in cells. Second, disorders in plastic metabolism. Third, damage to cellular and subcellular membranes. Fourth, disorders in enzymatic systems. Fifth, impairment of water electrolyte balance in a cell. Sixth, derangement of the genetic program of activity of cells. 7. Impairment of receptor apparatus of cellular and intracellular mechanisms. And number 8, impairment of cell-to-cell interactions. Let us look at some of them. Impairment of water electrolyte balance in a cell. It has the following kinds: increased or decreased amount of different cations, anions, and water inside and outside cells. Second type is hyper and dehydration of cells and extracellular structures. Let us look at an example. In the picture, you see changes in erythrocytes in solutions with different osmotic density. You see that in the hypotonic solution, the cell undergoes lysis, the phenomenon called hemolysis. Water in an organism moves according to laws of osmosis. It moves from the region of low osmotic pressure to the region of high pressure, in other words, by concentration gradient. In hypotonic solution, water enters cell, which swells and ruptures, the phenomenon called osmotic lysis. In the hypertonic medium, water moves by concentration gradient from the cell outside, and the cell shrinks, the phenomenon called plasmolysis. In the environment with normal osmotic pressure, the cell is not damaged. Cell damage may be linked with accumulation of oxygen-derived free radicals, oxidative stress. Free radicals are chemical species with a single unpaired electron in an outer orbital. They are extremely unstable and react with inorganic and organic chemicals. When generated in cells, they avidly attack nucleic acids as well as a variety of cellular proteins and lipids. In addition, free radicals initiate reactions in which molecules that react with free radicals are themselves converted into other types of free radicals, thereby propagating the chain of damage. In the picture, you see reactions of free radical oxidation. The first is formation of active form of oxygen, superoxide radical. The second shows that superoxide radical enters a reaction of spontaneous dismutation under action of superoxide dismutase enzyme. In the aqueous medium, it results in formation of hydrogen peroxide, which is destructed by catalysis. Number 3 shows that these active forms of oxygens may interact with each other and form free radicals. of which especially dangerous is hydroxyl radical, which provokes further damages in the cell. Reactive oxygen species cause cell injury by three main reactions. First is lipid peroxidation of membranes. Double bonds in membrane unsaturated lipids are vulnerable to attack by oxygen-derived free radicals. The lipid radical interactions yield peroxides which are themselves unstable and reactive, and an autocatalytic chain reaction begins. Second is cross-linking and other changes in proteins. Free radicals promote sulfhydryl-mediated protein cross-linking, resulting in enhanced degradation or loss of enzymatic activity. Free radical reactions may also directly cause polypeptide fragmentation. DNA damage. Free radical reactions with thymine in nuclear and mitochondrial DNA produce single-strand breaks. Such DNA damage has been implicated in cell death, aging, and malignant transformation of cells. In an organism, there is a kind of protection of oxidants. These are antioxidants, which may be enzymatic and non-enzymatic. Enzymatic antioxidants include superoxide dismutase, catalase, glutathione system, and thioredoxin system. Non-enzymatic antioxidants are vitamins E, A, C, coenzyme Q10, selenium, zinc, carotenoids, and bioflavonoids. Damaged cell possesses some mechanisms of compensation for damages. They are compensation of disorder in energy metabolism, protection of cell membranes, compensation of water-ion imbalance, repair of cell genome. In result of activation of this mechanism, in tissue or organ, they may develop atrophy, hypertrophy, hyperplasia, metaplasia, dysplasia. Atrophy is a reduction in the volume of organ. In result of reduction in the volume and size of each cell in the organ with the same number of cells. Causes: decreased workload, loss of innervation, diminished blood supply, inadequate nutrition, loss of endocrine stimulation, aging. In the picture you see atrophy of muscle fibers. Hypertrophy. Hypertrophy is enlargement of an organ in result of increase in the volume and size of each cell in the organ without increase in their number. Causes: increased functional demand, specific hormonal stimulation. Hypertrophy occurs under both physiologic and pathologic condition. In the picture you see hypertrophy of the left ventricle of a patient with hypertension. Hyperplasia is an enlargement of an organ in result of increase in the amount of cells due to their multiplication. It occurs in tissues where cells are capable of mitotic division, such as glands, bone marrow, tonsils, lymph nodes. In the left picture you see hyperplasia of tonsils. And in the right picture, you see hyperplasia of thyroid. Metaplasia is the phenomenon when one type of cells, epithelial or mesenchymal, is replaced by another type of cells. In the left picture, you see squamous metaplasia in respiratory tract of the habitual cigarette smoker. And in the right picture, you see intestinal metaplasia of gastric epithelium. Dysplasia is a deranged cell growth of a specific tissue that results in cells that vary in size, shape, and organization. Causes are chronic stimulation, chronic inflammation, precancerous conditions. And in the picture you see normal squamous epithelium and dysplasia. In the diagram, you see outcomes of cell damage. In case of insignificant damages, changes in the cell are reversible. In case of irreversible significant damages, the cell may die. Cell death is the final result of its damage. There exist two main kinds of cell death: necrosis and apoptosis. Necrosis is a pathological process induced by a pathogenic agent. Apoptosis is a physiological process based on the programmed cell death. Necrosis is the most common pattern of cell death. It may be defined as morphological changes that follow the cell death in a living tissue or organ and result in progressive degradative activity of enzymes. Two principal processes are responsible for changes in necrosis. First, enzymatic digestion of the cell, and second, denaturation of proteins. Causes of necrosis are identical to causes of cell injury in general terms, such as mechanical, chemical, physical factors, infectious agents, hypoxia, or anoxia. Morphological changes in necrosis are the following. In case of reversible damage, there occurs swelling of endoplasmic reticulum and mitochondria with probable appearance of membranous blebs. In case of progressive damage, there occurs rupture of the cytoplasmic membrane, of membranes of organelles and nucleus, breakdown of mitochondria of nucleus with leakage of their contents. This process is obligatorily superimposed with inflammation, with the result of alteration in the architecture of tissues. Morphological types of necrosis. Coagulative necrosis is characteristic of hypoxic cell death. The best example of coagulative necrosis is myocardial infarction. Liquefactive necrosis results from the rapid action of hydrolytic enzymes and always occurs when autolysis and heterolysis prevail over denaturation of proteins. It is characteristic of ischemic necrosis of the brain, pancreas. It is common in bacterial lesions where it occurs due to activity of bacterial enzymes, of bacterial and leukocytic enzymes. A good example of liquefactive necrosis is brain infarction. And here you see a picture of caseous necrosis. It is a type of necrosis that is a combination of coagulative and liquefactive necrosis. It is usually seen in tuberculosis. Fibrinoid necrosis is a type of connective tissue necrosis seen particularly in autoimmune disease when collagen and smooth muscles are affected, for example, in polyarteritis nodosa, when fibrinoid necrosis affects blood vessel walls. Fibrinoid necrosis is characterized by loss of the normal structure of collagen fibers. Gangrene. The term gangrene is commonly used in clinical practice to describe a condition when extensive tissue necrosis is complicated by bacterial infection. There are three major types of gangrene: dry, wet, and gas. Dry gangrene is the condition when necrotic tissue appears black and dry, and it is sharply demarcated from viable tissue. Most commonly, it occurs in extremities as a result of extreme ischemic coagulative necrosis due to arterial obstruction. When the coagulative pattern prevails, the result is dry gangrene, and when liquefaction is more pronounced, the result is wet gangrene. Apoptosis is referred to as programmed cell death because it happens due to biochemical instructions in the cell's DNA. This is opposed to the process of necrosis, when a cell dies due to outside trauma or deprivation. Apoptosis is stimulated by signal molecules that tell the cells that it's time to commit suicide. The two major types of apoptosis pathways are intrinsic pathways, where a cell receives a signal to destroy itself from one of its own genes or proteins due to detection of DNA damage, and extrinsic pathways, when a cell receives a signal to start apoptosis from other cells in the organism. The extrinsic pathway involves transmembrane receptor-mediated interactions. These interactions take place between ligands and their corresponding death receptors that are all part of the tumor necrosis factor family and called death domain. The death domain transmits the death signal from the cell surface to intracellular signaling pathways. As a result, a death-inducing signaling complex, DISC, is formed, resulting in the autocatalytic activation of pro-caspase 8. Once caspase 8 is activated, the terminal phase or execution phase of apoptosis is triggered. The intrinsic pathway involves a series of non-receptor-mediated processes that produce intracellular signals and act directly on targets within the cell. This pathway involves mitochondrial-initiated events. Here, changes occur in the inner mitochondrial membrane that cause the opening of the mitochondrial permeability transition pore and release of pro-apoptotic proteins from the intermembrane space into the cytosol. In result of a number of reactions, caspases are produced that translocate to the nucleus and cause DNA fragmentation and condensation of peripheral nuclear chromatin. Perforin granzyme pathway is a novel pathway employed by cytotoxic T lymphocytes that exert their cytotoxic effects on tumor cells and virus-infected cells. This involves secretion of the transmembrane pore-forming molecule, perforin, with a subsequent release of cytoplasmic granules through the pore and towards the target cell. The granules consist of two proteases, granzyme A and granzyme B. Granzymes activate caspases and the execution phase of apoptosis starts. Execution pathway is considered the terminal pathway of apoptosis. This phase of apoptosis is initiated by the activation of various caspases that activate cytoplasmic endonucleases and proteases, which degrade the nuclear and cytoskeletal material. Caspase 3 is the most important protein of the executioner caspases. Sequence of events in apoptosis. First event is elevation of cellular calcium and rapid reduction of volume of the cell. Then, activation of calcium-dependent enzyme endonuclease, which cleaves DNA. The third event is fragmentation of DNA and marked condensation of both nucleus and cytoplasm. It is followed by formation of apoptotic bodies. Small apoptotic bodies are composed of fragments of nuclei with condensed chromatin. Larger apoptotic bodies are composed of both fragments of nuclei and condensed cytoplasm with preserved organelles. Apoptotic bodies are rapidly phagocytized by neighboring cells or by macrophages. As the process takes place without a release of cellular components, no inflammatory response occurs. Here you see an example of apoptosis of human leukocyte. Significance of apoptosis. Apoptosis is essential during development, where many cells undergo programmed cell death, thus contributing to formation of various tissues and organs from a single mass of tissue. It can even be used as an important determinant of fetal abnormalities. The regular removal of old cells from the body enables the body to produce new cells, helping maintain the cell population in the body. The inability to do so might result in dramatic consequences depending on the type of cells. Apoptosis helps in the removal of redundant and damaged cells from the body. At the same time, cells that are infected with a virus and the ones that cannot be repaired are removed via apoptosis. Apoptosis is often used by the body's immune system to check if the newly formed cells are self-destructive or not. If the immune cells are found to be destructive for the body cells, they are removed, preventing the possibility of autoimmune diseases. Apoptosis plays a critical role in the immune system of the body. Different cytotoxic cells, like T lymphocytes, are produced in advance during infection by foreign materials. Once the invader is cleared from the body, the existing pathogen-specific immune cells are removed from the body through apoptosis. Apoptosis has also been observed in regulating the development of thymocytes and the shaping of T cells and coordination of various immune responses. Apoptosis is also involved in the removal of about 50% of neurons produced during early embryonic development and in the formation of reproductive parts. Excess apoptosis might result in neurodegenerative diseases, and deficient apoptosis occurs in conditions like cancer and autoimmune diseases. Here, please, you see the table which will help you compare necrosis and apoptosis. Thank you for your attention.